The overall goal of this procedure is to establish primary endometrial stromal cell cultures from resected hysterectomies. This is accomplished by first distinguishing and separating uterine layers to isolate a section of endometrial tissue. The second step is to choose a cell isolation technique between the scraping and trypsin methods. When utilizing the scraping method, mechanical force and scraping are used to separate and dissociate endometrial stromal cells. Alternatively, when utilizing the trypsin method, the enzymatic activity of trypsin is used to separate and dissociate endometrial stromal cells. Ultimately, microscopy is used to observe cell growth and morphology, while other techniques discussed in the manuscript are used to verify endometrial stromal cultures.
This method can help answer key physiology questions in the reproductive field, phenomena such as the reproductive cycle and menstruation.
Hysterectomy specimens used in this manuscript were collected in concordance with a university IRB-approved ethics protocol numbered IRB HSR number 14424. Maintain patient-derived tissue in a 50 milliliter tube containing RPMI or DMEM with high glucose at 4 degrees Celsius for no more than 24 hours before the sample is processed. When ready to begin,